The Human Genome Project, also referred to as HGP, is an international research effort to determine the DNA sequence of the entire human genome. There were several sources that contributed to this project, and they include the National Human Genome Institute, the US Department of Energy, numerous universities and labs throughout the US, and international partners in the United Kingdom, France, Germany, Japan, and China. The Human Genome Project aimed to explore and map the full set of genetic instructions on the human genome. The aim was to describe the exact sequence of the 3 billion nucleotide pairs. The Human Genome Project was completed ahead of schedule in 2003 due to unexpected advances in data processing technology. Not only did the Human Genome Project focus on humans, but also 5 other organisms including the laboratory mouse and the fruit fly. There were several techniques that were used and invented to make the Human Genome Project possible. These included DNA sequencing, YAC, PCR, BAC, and electrophoresis. DNA sequencing is the process of sequencing DNA and is basically determining the order of the 4 chemical bases that make up the DNA molecule. The technology of DNA sequencing was made faster and less expensive as part of the Human Genome Project. Yeast artificial chromosome is a human-engineered DNA molecule which is used to clone DNA sequences in yeast cells. YACs are frequently used in association with the mapping of genomes. Sections of an organism's DNA can be implanted into YACs. Yeast cells then take up the YACs with their inserted DNA. As the yeast cells mature and split, they intensify the YAC DNA, which can be isolated and used for DNA mapping. There is also a similar method, BAC, which stands for bacterial artificial chromosome. The difference between these two methods are: YACs are genetically engineered chromosomes derived from the DNA of the yeast. BACs are an engineered molecule used to clone DNA segments in bacterial cells. It's not hard to construct YAC, where it is relatively hard to construct BAC. YACs are unstable, whereas BACs are stable. Electrophoresis is a technique that is used to separate charged molecules such as DNA, RNA, and proteins according to their size. When an electric current is passed through the gel, charged molecules will move through. An electric current is applied across the gel, making one side positively charged and the other side negatively charged. The charged molecules will migrate towards the opposite charge. Electrophoresis enables us to distinguish the different lengths of fragments of DNA. The results of the Human Genome Project are thought to revolutionize healthcare. At the moment, there is more emphasis placed on treating disease rather than preventing disease from establishing in the first place. Because we can now map the human genes, there will be more emphasis placed on establishing the potential risk and preventing disease. This will in turn benefit as less disease will occur. The genome map will be used to study reasons for controlled cell division. An example of this would be in cancer. The genome map will be used to study over 4,000 hereditary diseases. Some other benefits revolve around forensics with DNA, identification of victims, the matching of organ donors and recipients, improved genetic counseling, risk assessments of mutagens, and the potentials of gene therapy. The Human Genome Project also provides detailed information which supports the understanding of human evolution and migration. Some limitations are only a simple representation of the relationship between genes and organisms is provided by DNA makes RNA makes proteins. Biochemical pathways shape a complicated network that is beyond merely reducing life to identifying individual genes located on chromosomes. A gene is far more complex than the data may suggest. This is because genes frequently include non-coding regions that regulate the gene expression. Areas called introns are detached from the messenger RNA in a method that's called splicing. Depending on how the gene is spliced, it may code for different proteins. A full gene, therefore, is much more than a coded set of guidelines for a protein sequence. The genetic coding for any individual appears to be interlocked with the coding of other genes. Each part depends on one another. More limitations are chromosomes contain numerous repeat sequences of DNA that look alike. The hard part is reorganizing the fragments in the order in which they occur on the chromosome, once the order of nucleotides has been determined. This includes doing a few things such as identifying overlaps, fixing any sequencing errors, and then filling in any parts of information that may be missing. After all of this has been completed, there is also the job of identifying the proteins that are used to make the human being. Just because we know the sequence of proteins doesn't mean we will be able to determine their function. And in order to determine the amounts of RNA each gene is and isn't producing, scientists will need to map and edit the mRNA. Genetic mapping is one of the tools that the Human Genome Project established to recognize new genes and understand their function. Genetic mapping can show proof that a disease that has been transmitted from a parent to their child is linked to one or more genes. Not only does genetic mapping show this evidence, but it also offers clues about which chromosome holds the gene and specifically where the gene lies on that chromosome. Alfred Sturtevant created the first genetic map of a chromosome from the fruit fly in 1913. He projected the idea that the frequency of crossing over between genes could perhaps help determine their position on a chromosome. By finding out how frequently several characteristics are inherited together, it's possible to estimate the distance between genes. A map of where the genes are in relation to each other on the chromosomes can be made. This is known as a linkage map. This same idea affected the development of the first human genome map. If two or more characteristics were seen to be regularly inherited together in a family, an example of this would be blonde hair and blue eyes, it is implied that the genes for the two characteristics were near each other on a chromosome. Reasons why linkage maps couldn't be used to map the human genome are specifically due to the accuracy of the linkage maps not being good enough. Linkage maps are entirely based on the recombination percentage, which only really provides a relative position. Linkage maps were also not used to map the human genome because they have only been used to map genes with visible characteristics, and the human genome is 99% non-coding, which basically means there will be very little visible inherited genes. As mentioned earlier, linkage maps were made based on experimental cross-testing of the fruit fly. Therefore, they are not suited to human beings.